Okay, so we're going to go on from what white matter and gray matter are onto synapses between neurons. Also talk about graded potentials and action potentials. So a synapse is a gap between neurons or between a neuron and a receptor or effector. So you have a neuron cell body with dendrites, and then you have the axon coming off, and it goes down here, and say it has a couple axon, a couple terminals, and this one hits another. Oops. Hold on a second. So it goes on, and there's another neuron cell body with some dendrites. Okay, and so this little gap right there is the synapse. Okay, this is a synapse between two neurons, but this could just as easily be a motor, like a skeletal muscle cell, or a gland cell. But that gap is a synapse. So where two things come, two neurons or a neuron or receptor or effector come together. If a neuron sends a neurotransmitter, it is called the presynaptic neuron. So this would be that right there is the presynaptic neuron, and this is the postsynaptic postsynaptic neuron. So the neuron receiving the neurotransmitter is the postsynaptic neuron. So when the neurotransmitter goes across the gap, the postsynaptic neuron produces a type of potential. It's called a graded potential, called a postsynaptic potential. Postsynaptic neuron converts it to an electrical signal. So there are three types of neuron-to-neuron synapses. There's axo-axonic, axodendritic, and axosomatic. Axon to cell body. So you can have so an axon can communicate with another axon. It can also communicate with a dendrite or directly to the cell body of a neuron. But you notice they're all axons communicating. So the axon is going to be the presynaptic part. You can have an axon be the postsynaptic part. You can have the dendrite be the postsynaptic part, or cell body be the postsynaptic part. A neuron to muscle synapse is called a neuromuscular junction. Next slide. Okay, so here's showing a diagram of some neuron to neuron synapses. So here would be an axon to axon one here, axo-axonic. So here's an axon, signal comes in here, and it touches the actual axon hillock portion, but it touches here. So it starts an action potential in the axon hillock, which then would help fire this axon or this neuron. You can also have an axodendritic, so an axon to dendrite. And so the dendrites are the postsynaptic part. This axon will obviously be presynaptic. And then you have an axosomatic. So you have axon is the presynaptic part, and the soma, the cell body here, will be the postsynaptic part. So that's axosomatic, an axon to the soma, the body of the neuron. And then when it triggers, what happens is there's what's called a graded potential. Okay, graded potential, and this graded potential is a collection of electrical charges that travel down towards the trigger zone. Okay, and when they collect down in the trigger zone, or in the axon hillock and the trigger zone, and then when it gets to be enough graded potential, then it fires, and you send a signal down the axon, and it's called an action potential. It's called an action potential because it has a potential to do something, a potential to have an action. And then when it reaches the axon terminal, then you have a release of neurotransmitters are released across the synapse, and those then innervate. They cause a muscle to contract. They cause a gland to release a hormone. So then that's how that works. Next slide. Here's what a chemical synapse looks like. So we have up here, we have the larger view. We have the presynaptic neuron here with a postsynaptic neuron here. So here's the neuron cell body, and so this is a axodendritic. Sorry, I can't write that well with this. I'm using a pad. Previous ones I wrote on the screen. I'm having a hard time writing on this pad, or very well. So hopefully I'll get better at this. So we have a presynaptic neuron. So this is axodendritic. It's an axon to dendrites. So then that causes these graded potentials to collect down here in the axon hillock region. And then when enough of those collect, it fires, and you get an action potential go down the axon. When it gets down here, and it goes down here to the end, that's what this green is. You have this this action potential comes down here, and it causes calcium to go into the cell. So calcium goes in, showing the arrows here. And there's neurotransmitters that are inside these synaptic vesicles, if you remember them from studying the cell a couple weeks back. So there's neurotransmitters that are inside those vesicles, and those vesicles go and they fuse. Due to calcium content, they fuse with the cell membrane, and they release the neurotransmitters, and those go across this gap. They go across this gap from this exocytic synaptic vesicle, and then they collect over here, and they activate a neurotransmitter receptor, which then brings more of those, brings some ions, allows some ions to flow into and trigger the second axon. So this here was the presynaptic neuron or axon, and this is the postsynaptic neuron, and the specific part of the neuron is a dendrite. It's what that axon sent those neurotransmitters across to activate. Next slide. Here's a electron micrograph of a presynaptic neuron and a postsynaptic neuron. And you can see the presynaptic neuron there, and then the postsynaptic neuron here. So and the gap right there between the two things, it looks like it's touching. There it is, going to be a really tiny little gap there, is the synapse. Next slide. So with a neuromuscular junction, this is a spinal reflex. So you put your finger next to a flame, ow, it hurts. So the direction of the impulse, it goes up a sensory neuron, okay? And it goes, so you have pain receptors in the skin here, okay? And so the flow of the impulse goes up the sensory neuron here. It goes up to the spinal cord. And then you have the dorsal root ganglion where the cell body of the sensory neuron is. And it continues to transmit the signal up to the spinal cord. Spinal cord sends a signal back down the motor neuron, the blue one, down to the muscle, and it activates the muscle to pull your hand back. You might also send a signal at this point, instead of just sending a signal down here to pull your hand back, you might also send a signal up to your brain saying, hey, I've touched something and it was really hot, and I might want to get out of here because there might be other hot things around. So it doesn't have to just be this one little pathway, but this is a spinal reflex. Would be you feel pain, you pull it back, pull your hand back real fast. Next slide. Here's another neuromuscular junction. We talked about this one before, where you have a sensory receptor feeling the pin between your fingers, sensory neuron going up to the spinal cord, then a signal goes on and continues onto the brain. The brain collects information, sends information back down to the various muscles to fire, and fires those muscles and makes those muscles contract and move the pin. Next slide. So synapses, summary, they're a junction between two neurons is a synapse. You have a synaptic cleft, a gap separates the two neurons. You have presynaptic neuron and postsynaptic neuron. If the presynaptic neuron reacts with a muscle gland, then that cell is called a postsynaptic cell. And junction between a motor neuron and muscle fiber is called a neuromuscular junction. And you have acetylcholine, which is often abbreviated ACH. Acetylcholine is the neurotransmitter. Synapse between two neurons, the neurotransmitter is any of several possibilities that open up the ion channels. Synapses are gaps between myelinated cells, between myelinated neurons. This is different from the nodes of neuron VA, which are gaps along the axon between the Schwann cells. Next slide. Okay, so what is a graded potential versus an action potential? Graded potential is a collection of electrical charges at the dendrites. Action potentials are at the axon. So what happens is you collect a whole bunch, and these are graded potentials, so you get a bunch of potential, and what you have is a collection of charges. So you have some plus charges, so it actually be minus charges, and plus charges out here, and plus charges. And the more you get these minus charges collecting down here, then all of a sudden it causes some channels to open up and allow ions to flow back and forth across the plasma membrane to neutralize that charge difference across the membrane. And so what happens is you have continuous conduction. If you don't have these are Schwann cells here, 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 and here. If you don't have these, and here, all the way down. If you don't have the Schwann cells, then you have unmyelinated axons. So you have continuous conduction of these charges all the way down the axon, and it's going to be very slow. But if you have the Schwann cells, what it does is it wraps up the Schwann cell around the axon so that these ion channels can only open up here at the nodes of neuron VA, there, there, and there, and there. So that means that the only time that this charge gets the charge difference gets neutralized is at these nodes of neuron VA. So this is called saltatory conduction, which is from Latin saltare, to hop or leap. And so it doesn't really leap, but it's the fact that the charge is these sodium and calcium channels open up at each of these nodes of neuron VA along here. Next slide. So now we get into a little bit of math. Well, it looks like math. It's sort of math. It's not really. So, but you just have a graph. So what you have are called a membrane potential. And typically your membrane potential is about minus 70 millivolts, okay? And you have a small stimulus like this, or like this, or like this. It's not enough to trigger depolarization of the cell membrane. What you have is cell membrane. You have plus charges out here. You have minus charges on the inside. And so you have what's called polarity, which means it's like battery poles. You have plus poles and minus poles on a battery. So you have different charges. So if you depolarize, that means that these charges mix back and forth through ion channels that wrap back and forth across this membrane. And then you can depolarize the membrane. So, but if you have enough of these charges collect, then you can have a large stimulus, and that will cause complete depolarization, and then you fire. And so what happens is you fire this nerve impulse, and it starts going down. It essentially goes down the axon. And then you have what's called a hyperpolarization, because there's other channels that open up and start shifting these ions back and forth across the membrane. So you actually go down past minus 70 to almost minus 85 to minus 90 millivolts, and then you come back. Everything comes back to normal, and it comes back to the minus 70 millivolts, which is where you want to be in terms of the polarization of the membrane. Next slide. So here's a way of measuring this. You can actually put a little probe inside the cell, and you have the minus charges inside the cell and the plus charges outside the cell. You can measure that with this guy, and the difference is minus 70 millivolts. This is ACF is extracellular fluid. That's a plasma membrane. Here's your cytosol or cytoplasm. And so here's a gated channel. It's called a gated channel. that won't let these ions go across until it gets opened up, until it's stimulated to open up, and then allow those ions to go across. So this is a resting state. Next slide. So you have a threshold here, and you have failed initiations here. So it's not enough of a change in the membrane potential to fire, but then all of a sudden you have a big stimulus here, and it fires, and it goes up. So it depolarizes, then it starts repolarizing. So you have sodium ions flow into this you have this ion channel here. So sodium ions flow into the cell, and so you have all these minus charges here. And so all of a sudden you get a whole bunch of plus charges inside from sodium ions, and it starts neutralizing this. So your potential actually, and there's so many of them flow in that this actually becomes positive on this side, and this becomes slightly, relatively negative. So your membrane potential changes from minus 70 to plus 40. Then other channels open up, and they start pumping actually calcium ions back out, as well as sodium ions back out to try to repolarize the membrane. And they keep pumping, and then they realize at one point, oh, we went too far, so we have hyperpolarization. So then they pump some of them back inside the cell and some of them back outside the cell. Then you get back to your minus 70 millivolts. And this the section through here. So once you depolarize, you've fired a nerve impulse. This whole section through here, is a refractory period. Refractory period where the nerve impulse, the nerve cell can't fire again. So it always has to be so the nerve cell has to be kind of careful about when it fires, because it will be unable to fire again immediately. It has to repolarize, and then it can fire again. So it does take a little bit of time. Next slide. So here's a figure from your guys' textbook. I think I have this broken down into a couple different pieces. Yeah, I did. So good. Okay. So here we are. This is all in the trigger, what's happening in the trigger zone here. So you have the potential, a local potential comes along, you have some plus charges, minus charges, and so all of a sudden you have a sodium channel and a potassium channel, and the sodium channel opens up when these charges come along and allows all these positive ions to flow in, and this makes it all positive. These positive charges notify this calcium channel to start opening up, and it'll start sending some positive ions back out. Here in a second, we'll see the next slide. So the calcium ions start getting pumped out. The sodium channel closes. The potassium ions are flowing out, and so the depolarization, it starts repolarizing. So it starts going back down this way, and then you end up hyperpolarizing down here. So you have a few too many plus charges out here and a few too many minus charges down here. So then you have to pump a few plus charges back in, a few minus charges, a few plus charges back in to neutralize those minus charges to get back to your minus 70 millivolts. So you have the whole refractory period is really this entire thing here. You have an absolute and relative refractory period where you can't fire twice during this period. You can't fire again. It's already in the process of firing or has fired, one of the two. So next slide. And so here's just another picture of the sodium ion channels where the sodium ions flow in, and as the plus charges come along and the membrane is depolarized, it opens up more and more of those sodium channels. Next slide. Okay, so the last thing we would be doing would be observing a neuron smear, looking at neurons and neuroglial cells, looking at posterior root ganglion. But you've basically seen that in the slides in this presentation. So you've seen those two things, and you also look at peripheral nerve. you'd see nodes of rod, VA, and fascicles, and epineurium, and perineurium, and endoneurium. So look at those pictures back in the PowerPoint as well. Make sure you know what those look like. So let's see. So the next thing we're going to go to is going to be the spinal cord, and I'll record that video next. And so we'll see you next time. Bye.